Well, working with Uwe for quite a few years, I'm pretty much used to see last-minute slide optimizations. It's a new level now here. It's on German, yeah, it's true, it's my laptop, so it has to be on German. That looks like it's getting started. So the new level of last minute slide deck preparations was defined by Phil Doherty, who is our next speaker. Phil began his PAT career over 3 decades ago, and 21 of those these years he was working with Pfizer R&D and manufacturing, delivering tech that is now common in today's PAT world. I know personally that he was working on blend uniformity a long time before it was started to be shown on conferences. When he left Pfizer, he was studying his consultants business as process analytics, something, whatever. Then he joined Expo Technologies, where he was a managing director, and he recently screensaver mode. That's about the last minute BIOS. And he's now founder and CEO of Acuitis Process Analytical Technologies, develops total PAT solutions suitable for use from lab to proof of concept, all the way to regulatory filing and plant deployment. Great to have you here, Phil, as usual. And as often, stage is yours.
Thanks, Sebastian. Yeah, it seems like a long time ago that 2009 was the first time I came to Dresden to one of the Sentronic PAT. I always correct myself, PAT meetings rather than user meetings, because there's a wide selection of people who actually come to these meetings, and it's always been a great forum for discussion and to talk about all sorts of PAT, not just near-infrared and not just Sentronic as well too, which is one of the great things about this meeting. So today I want to continue the conversation that Manuel started about blend uniformity and online blend analysis. It was a great introduction. First of all, shamelessly, the people that we work with over the past decade or so is everybody, basically. As Sebastian mentioned, we do all sorts of PAT, and not just in pharma, but also in food as well too. So for today's agenda, I want to talk about some of the background of the application of near-infrared blend analysis and its development, how we can develop rapid quantitative methods, how we can approach endpoint determination, and importantly, how we can use the tool to understand badly behaving blends and formulations as well too. So in concept, it's fairly simple. We take raw materials and we add our API to it and we mix them, and hopefully we form a uniform mixture that's suitable for safe and effective and efficacious medicines for our patients in the end. There's a slide here that I've stolen from Steve that he might use again later, but the important thing to understand is that blends often don't like to behave well, you know. Within blends, we have some intrinsic material properties where the API likes to associate with some of the excipients selectively, and materials like to clump and make formations within the blend that are difficult aggregates to break up and can affect our final dosage form performance. And here I like to use this illustration, which is an electron micrograph of a blend, and you can see that in this example, the large particle is the disintegrant within the tablet formulation, and the API has selectively coated the surface of the disintegrant particles themselves. So a little bit of a time machine step back. The first application of near-infrared blend analysis really dates back from about 1992. So there was a guy at Perstorp, and at one stage, Voss Perstorp was the only game in town for near-infrared analyzers, and he published an application note where he took some samples, measured some blend at different time points from a blending process, and showed that you could measure changes in the near-infrared bands as a result. Hot on the heels of that, at Pfizer, we took a Dewar flask, which was the handiest stainless steel container we could find. We cut a hole in it and put a sapphire window, and then just tumbled it and then stopped and held a probe up against the window to measure the blend inside the actual container itself. Once we proved that that worked, that was rapidly changed over to an at-line method where we could measure blend top, middle, and bottom in an intermediate bulk container. And that sort of set the scene then for the development of the original automated system. Again, at the time, we only had access to very slow scanning instruments. It took about 30 seconds to acquire a near-infrared spectrum on a FOSS 6500. And so what we did was put the fiber optic probe straight through the axle of the blender and introduce the probe into the actual blender itself. This is proof from yesterday when we had the workshops. Uwe suggested that we separate on hair color, and I did used to have hair at one stage when I started doing PAT back in 1993. But this resulted in the development of a system very similar to the concept that Mano talked about there, where we could change the geometry and scale of blend vessels from 25 milliliters up to about 10 liters was the biggest size that we could go in there. Off the back of that, we could introduce real-time analysis. So this was very important. Up to that point, you had to take your blend near-infrared spectra, take them offline, do some PCA on them, and typically at that stage, you had the introduction of modern, what's currently desktop PCs, and it used to take about 2 hours for a PCA calculation on a spectral data set of about 10 spectra. It took a long time. So what we developed was a real-time system that could do things like some model-free approaches that Adam talked about yesterday that hopefully will come again up in conversation. But the idea of using a target, which I'll talk about, is we rapidly move from this sort of resolution to having more frequent sampling, and then eventually to what we know today of a measurement every rotation in real time from the system. As I mentioned, some model-free approaches were developed at the time using a thing called dissimilarity, which has sort of gone out of fashion but was useful at the time, which gives an indication of not just the rate of mixing, but also this is using a target of 2 of the raw materials in the blend, the API and the major excipient, and looking at the overall distribution and breaking up of aggregates and lumps to form a uniform blend at the end. Straight off the back of that, the big boys wanted this to go into production, so we had to work at technology that would actually move the measurement point away from a spectrometer on or near the actual blender to something that was mounted onto it. So this idea of having a remote reflectance head and a piggyback system, which we're all familiar now with, was back in 1997. We built this system. So it's a long history of the application of online blend analysis. Just for people to understand as well too, some of the things that are interesting about near infrared is obviously we're looking at reflectance spectra, and the bed density and compaction and the particle size all play a part in the near infrared response that we get. Basically, as we get less dense and larger particles, we see a baseline offset that can be very, very useful. Some of the things that formulators care about is not just the potency and the content uniformity, but also the flowability and the compaction behavior in the actual blend itself. So thankfully, people like Sentronic came along and gave us a very elegant solution of battery-powered spectrometers that could be used for this application. Prior to this point, you had to power the spectrometer through a thing called a slip ring and pass communications back and forth to the rotary side of the tumble blender as well too. But now, these days, we have, let's say, much nicer solutions like the BU2 and the DA unit, which is the more recent iteration of the online blend analyzer from Sentronic. So, in concept, what we can do is provide continuous information. As Manuel mentioned, we can optimize the actual blending process itself with a really good indication of the endpoint in the blend and also the stability of that endpoint and how good of a mixture we've actually made. Don't have to talk about this really too much. What is interesting is that the technology now has got very accurate, I think it's in 9-axis sensing on the actual devices themselves. So you've got a really good control of where you acquire your blend at the bottom of the rotational cycle. So in effect, we've got a static sample in front of the analyzer, and so we can track very accurately different bands within the spectral range. So we can track things like dispersion of mic stearate. Magnesium stearate has got a lot of CH2 units in it, so it's got a massive near-infrared response, so near-infrared is very, very sensitive to it. And we can also track the dispersion of the API and the behavior of the excipients as well too. This is also an important aspect, especially from a formulator standpoint, is to understand that stability and dispersion mechanisms within the actual blend itself. So in essence, we're measuring a sample that's static in front of a sapphire window. In something like sorry, there's a typo there. In the BU2, you've got a 12.7 millimeter spot size, and so by the time we do some calculations on the size of the cylinder of material that we're actually interrogating, we're actually measuring about 127 to 130 milligrams of material. So it's getting towards a dosage unit equivalence, which is the ideal really for PAT measurement in solid oral dosage forms. So in concept, what we have is a snapshot on every rotation as we move towards a blend endpoint, and that gives us a really good insight into what's going on inside the blender itself. So very quickly, I want to talk about efficient ways of building calibrations. Mano gave a great example there of using micro blends, but you can also use macro scale, small scale blends as well too, to build PLC models. And this idea of material sparing approaches is very, very popular within development, mainly because the cost of API is so outrageously expensive, and or it's just not available early in development. And so when your colleagues and your customers are pushing for effective manufacture of clinical supplies, especially, you know, you're the person in the spotlight, and you've got a good story to tell. But again, as Mano mentioned, everybody wants an API concentration and not just an arbitrary score value. And so what we can do is do a very, very quick stepwise addition of API and then redilution with addition of various excipients to work up and down a concentration gradient. And just to show you one of those stacked, this is for a dual API formulation of 2 granulates that we used in an animal health product. And here we can see the two mixing profiles and peaks that we're reaching for the 2 APIs, and get a nicely developed PLS model for both APIs in a very short set of experiments using a small amount of material. So moving on to blend endpoint determination. You know, say the systems are capable of very, very sophisticated endpoint determination. Usually we're just looking at the rate of change in spectra against a predetermined set point value. And there's a couple of tools that can commonly be used, the main one being the moving block pool standard deviation and the moving block F-test for looking at the rate of change and stability of the blend in the end. I mentioned PCA is a useful analysis tool, mainly because the different score values will typically tell you not just what's going on with the API distribution and dispersion processes within the blend, but also the way that the excipients are mixing as well too. And it's probably, if anybody's worked in this area, it's no surprise to know that often the dispersion of the different ingredients within a blend actually happen at different dynamics and different rates as well too. So you might have a very nice uniform content uniformity from the perspective of API concentration, but a functional excipient within the blend itself can also not be dispersed correctly, and you can track the dispersion of those sorts of things using PCA. So moving block standard deviation is one of the first approaches that were actually used within endpoint determination. And the way that works is basically we take spectra from the blender, and with each rotation we add spectra to the block to a predetermined size. And all we're doing is basically calculating the standard deviation of all of the wavelength absorbances and making a pooled version. And then what happens then is once we reach a block of 10 spectra, we inch along in an inchworm kind of way, and add a spectrum to the block and take a spectrum out of the block. And that allows us to actually produce a standard deviation plot that looks pretty much like this. So we don't have any data for the first part of the blend until we actually form a block. And then as we move through the actual blend process, we'll get an evolving value that's a very simplified singular value. We can also detect things like aggregate breakup and lump breakup, where you have a transient excursion from your target API concentration to a slightly higher concentration. And then as that material blends in, again, we can detect it quite readily as well too. So here's an example of looking at myxteriate dispersion and looking at two different block sizes. What's noticeable about this is this is a very well behaved blend, which is no surprise because the role of myxteriate in the actual formulation is to enhance flow characteristics. So typically you will see blends that when you go into the lubrication step, they become very stable and they disperse very, very quickly. One thing that is important is that over-lubrication is something that we should care about because it'll impact dissolution performance. Myxteriate is actually quite a nice waterproof jacket that can be put over every particle, and if you over-disperse it, you will impact your dissolution performance. So the second approach is a moving block F test. Again, it's a similar sort of approach, except that we're using two adjacent blocks of data in this case. And so as we form the blocks, we make an F test between the two populations, and we compare them for difference, basically. So again, we get a very similar response in that we don't, in this case, we have a longer time before we form the two blocks to compare, and we get a similar sort of outlook. Now what is important, and I hope this is probably not going to display very well, this was work that was done by Ad Gehrig and his group at Organon a long time ago, but it was an important consideration because you can see here the difference in sensitivity between a moving block standard deviation and a moving block F test. The F test is a much more sensitive instrument for detecting differences in dispersion in the actual blend itself. And if people are interested, I can dig out the reference for this paper as well too. It's back from 2016. But you can see here clearly where the F value is detecting much more sensitively the changes in this part at the end is the lubrication step in the blend compared to what's detected by the moving block standard deviation test itself. So on to how we can use online blend analysis as a diagnostic tool. An important consideration with near-infrared spectroscopy is actually the depth of penetration of the actual radiation itself. And we've got two things to contend with here. One is the fact that near-infrared absorption increases with wavelength. So what we get is a probability field of measurement, and the longer wavelengths penetrate further into the sample the shorter wavelengths penetrate further into the sample than the longer wavelengths. And this is something that's important to consider. So in reality, when you do studies on this on a typical blend, you're interrogating about somewhere between 350 and 500 microns of depth of material. So things like spot size and that depth become important. And so what that results in is if you have large aggregates or lumps within your blend, the occurrence of them within that measurement field or that probability field will give you an indication of either being accurate, an underestimation, or an overestimation of the actual content uniformity in the actual blend itself. So what that looks like in reality is here's a PLS output. Again, this is from Ad Gehrig's group, presented back in 2016. And it's a really nice example, and I haven't really seen a better example really of illustrating where we've got a PLS model running and indicating the concentration of the actual API in the blend. And here we've got a bad actor of a blend where you can see that there are large spikes in the blend. And the reason for the spikes is that as you have large lumps of API that come into that measurement zone, then there's an overestimation of the concentration. And you see overall, it's perceived as a low potency blend. If you took all of these points and took the average and take all of these points and take the average, they're overlay. So at a very bulk level, you have the right concentration, but there's clearly a problem with content uniformity. And they took the trouble to actually sieve these blends because they were investigating this bad acting of the API in this case. And here you can see in the case of the in the case of the bad blend, they had aggregates as large as 5 millimeters within the actual blend itself, which explains why you're getting these super potent spikes of really high concentration of API. So, other things to consider within blend analysis is that you'd be surprised how many formulators will try and stress their equipment by saying, well, we've got a blender of this volume, and we're going to put material in it that's not actually enough to fill it to an effective level. And sometimes what you'll see is that slumping occurs within the actual blender itself, and so there's an absence of shear force within the actual blend, within the vessel, and sometimes you'll get incomplete coverage of your window if it's been set up poorly with the timing of that measurement zone on the bottom of the rotation. So this is an example of a slumping blend not covering the window correctly, and here we can see a very large range of absorbances and a pretty poor spectra that's acquired as a result. Now luckily, these days we have really good filtering mechanisms. We can use things like dissimilarity measurements, or we can use within the Sentronic technology itself, it's got built-in bad scan rejection mechanisms. But you can clean this data up and get something usable out of it. So here's an example of applying joint time frequency analysis as a wavelet filter. In this case, that's been applied, and it's cleaned that data up enough to be usable. You know, not perfect, but we can still see the mixing dynamics and the endpoint occurrence within the actual blend itself. So here's another example of somebody stressing their equipment. Here we see a blend profile that's not typical of what we would like to see, which is a nice sort of second order mixing profile. Here we've got what's called diffusive mixing, and it's a lot slower process and causes problems. So quite clearly you see these blends are not reaching the final endpoint and are pretty poor. And if we overlay those and make a multivariate statistical process control chart, we see that this blend is nowhere near, it's just about getting to a plateau in this case. It was a bit of a near miss. This part here is the final blend step in this system as well too. And we can see that it's nicely reproducible. You know, you can't fault these people for flogging a dead horse. They did try a lot to try and make this work. But again, it's an indication of, you know, you can use this information to inform the formulator and the process developer to really help them on their way. And eventually they improved it, and eventually they got to the point where they had an effective mixing process. Once they, you know, surprise, surprise, they got their fill level right and got some shear happening in the actual blend itself. So one of the approaches that we used to use a long time ago was this representation, which I think is nice of using that moving block approach, where you can see the actual change in homogeneity as well too coming in. Again, it's an incomplete blend from one of these cases, but it's a good indication of overall content uniformity, a way of visualizing it as well too. So just in conclusion, online blend analysis can be more than just about content uniformity. It can be extremely useful diagnostic and development tool for use in development. Really important in scale-up, tech transfer, and process validation, and as a tool for continuous quality verification as well too. So just by acknowledgements, Steve Hammond works with me as my partner in Acuitis, and so we sort of wrangle all manner of PAT problems. A lot of this data came from Pfizer, from the guys who are now ex-Organon but now work at in-process LSP, Uwe and Owen from Sentronic, and the tools that we use from Brad at KAX and from Johan at Sartorius as well too. So thank you, and I'll answer any questions.
Thank you, Phil. Great to see all those example plots, and always excellent to see this pre and combination of PET and process understanding. So thanks a lot. We have time for 2 or 3 questions. If there are any, Owen is starting. Are you not going to claim your wrong last name? Or it's a question.
Sorry.
Great talk. On the note of seeing lumps in blend, one question I hear a lot from customers is, what's the right size for the NIR spot size for blend analysis because you're going to get more sensitive to scale scrutiny at a smaller spot. Any thoughts on kind of like what the magic spot size ought to be?
The ideal really is to strive. It's a double-edged sword, really, Owen. Ideally, you want to strive towards unit dose equivalence. So you want to sort of show, if this was a tablet or a capsule, this would be the variability on a dosage unit to dosage unit. But there is important micro information in there as well too. And it's important not to throw the baby out with the bathwater, really. When I worked at Pfizer for many years, we used to joke that they only had one process, which was blend, screen, blend, direct compression. They wouldn't touch any or granulation or raw compaction or anything, But that screening step usually should take care of any lumps and clumps, usually. But they can form post-screening as well too, as I mentioned, association and aggregate formation just between, particles that like to stick to particles that are their best friends, and some of them, if you've got a bad API, it likes to stay away from everybody and will top out and segregate. So it's really useful information. So.
Any other questions? No, looks like everyone needs a coffee now.
Yay, coffee.
Yeah, we shared the program on the table, so everyone is aware about the coffee break now. Thanks again, Phil. And as said, we have now a coffee break. Should be outside. I ordered a second coffee machine. They couldn't promise it for this break, so let's see how long the queue will be. See you here in 30 minutes. Thank you.